I want to thank all of our supporters, especially our Middlesex County freeholders and all the freeholders across the state of New Jersey, because we couldn't be here as extension agents and specialists serving you if we didn't have the support from the counties and from the state, and especially our support from the New Jersey Ag Experiment Station. So they make all the research and programs that we do across the state possible. Our presenter tonight is Dr. Tom Orton, and my colleague Peter Nietzsche is also here to help us answer some questions with genetics. The rest will do our best, but we're glad to have Tom on. He's going to give a nice overview of the history and basics of some of the breeding that's been happening at Rutgers, including our famous Rutgers tomato and asparagus. We've had some pretty outstanding varieties coming out of Rutgers over the many years in the past, including tomatoes and asparagus and blueberries and cranberries and strawberries. So we've got a long history of doing a lot of breeding work, and we've actually trained quite a few breeders around the planet to help everyone. But Tom has been teaching at Rutgers and doing a great job. He attended Michigan State University, in biology with honors and a PhD in botany and genetics. In 1995, he joined the faculty of the School of Environmental and Biological Sciences at Rutgers University, New Brunswick. Dr. Orton has taught plant breeding and advanced plant breeding courses at Rutgers University since 2003. He's co-authored over 50 research publications and 20 monograph chapters, a textbook for cultural plant breeding, and was a coordinator for 8 annual editions of the Mid-Atlantic Vegetable Production Guide, which is a huge benefit to all of our farmers. And without further ado, because we're running a little late, I'm going to turn it over to Tom, and we're going to jump in occasionally, Tom, with maybe a question here or there, just some comments. Maybe you could start off by just telling us a little bit how you got interested in plant breeding and then kind of work into your slides.
Well, how did I get interested in plant breeding? Well, when I was going to Michigan State long ago, the project that I was doing was a project in cell and molecular biology that where I was working very closely with plant breeders, and that's what really got me interested in plant breeding, was working with these guys and spending time in the field and seeing what they did and how they did it and why they did it. And so when I graduated from Michigan State back, it was a long time ago, late 70s, I took a job at UC Davis as a plant breeder. And although at that point in time I had never taken a plant breeding course, so it just goes to show you that sometimes taking a college course isn't the end all and beat all. You can be whatever you want, even if you don't take the class. So and I ended up just kind of learning on the fly as I went, and over 40 years later, I guess you learn enough to be called a plant breeder. So that's how it happened. And so I've been at Rutgers, as Bill mentioned, since 1995. The first 7 or so years, I was an administrator, and it hasn't really been. It's only over the last 15 years that I've really been working more in the area of plant breeding since about 2005. And so I've been working since then mainly on tomato breeding, a little bit on peppers, and kind of forays into some different crops since then. But through this career, I've been involved with plant breeding in a lot of different vegetable crops, including cole crops, broccoli, and cauliflower, celery, carrots, peppers, a little bit of asparagus. So it's been and eggplant as well. So,
and you've got some exciting new varieties. I know you're going to talk about today with your tomato. You're working with Pete and colleagues, and got a new exciting asparagus, a variety too that's coming out. So I can't wait to hear about that.
All right, let's move on to the next slide then. So what I found in my class that I've been teaching for 17 years now, and actually also at UC Davis, is that the best way to teach plant breeding really is to give examples of how it's done, and I decided today to take the lecture from a class that I actually give in the plant breeding class to compare two different vegetable species that are quite different, actually. And by seeing how they're similar and how they're the same, it goes a long way towards, I think, communicating how plant breeding works in a vegetable crop. It's a little bit different than how it works in an agronomic crop like corn and soybeans. So we're going to talk about asparagus and tomatoes today. All right, so plant breeding, what is it? Well, traditional breeding, and that's the title of the talk actually, is traditional breeding is the definition that I came up with is heritable changes to populations of plants resulting from the management of natural mating and selection. So plants are doing this all the time. They're mating and they're undergoing natural selection in natural habitats. And so humans come in and they alter that. They alter who's mating and how they're mating, and over many different cycles of making those changes, there are genetic changes that can occur if there's a heritable basis to what you're selecting for. And so that's traditional breeding, and it's been going on since the beginning of time for humans, ever since humans started farming. More recently, as you see in the bottom of the slide, the technical advances have been kind of plugged into the whole plant breeding platform, including things like induced mutations from chemical agents and radiation, soma clones from tissue culture, transformation with agrobacterium, so you can take any DNA sequence and plug it into a plant, and more recently this genome editing that you may have read about, CRISPR-Cas9, where you can go in and change any DNA sequence in the genome in a desirable way. And those are all used by plant breeders now.
Where you have there, we were talking about agrobacterium. Do you want to just mention how that's a natural process that occurs and does modifications within the plant?
Sure, yeah. That's the technology that has been around for probably about 35 years, and in that it's possible to isolate genes out of any source from nature, or actually to synthesize a gene and to put it into, to splice the gene into what's called a TI plasmid. It's just a circular piece of DNA that is bacterial, and that the function of that plasmid is to allow this bacterium, Agrobacterium tumefaciens, to invade the plant genetic apparatus. So if you hitchhike your gene onto the normal process of the pathogenesis of this bacterium, the gene actually goes right into the genome of the host. So what plant breeders have done and microbiologists is to disable the pathogenesis part, and then to use the agrobacterium just to put genes into plants. And commonly, these are called GMO. So there's some stigma attached to that. Of course, GMO has been around probably since the early 90s, and there's been a stronger reaction against GMO in probably Europe than in the North and South America. But there's still a lot of pushback on GMO. Does that explain how agrobacterium works to create GMO crops?
Yes, I was just trying to emphasize that crown gall or Agrobacterium tumefaciens is a very common thing found in nature. So it's nature's way of kind of manipulating the plant to produce proteins that the bacteria feed on. So in other words, plants do this, or bacteria and other organisms manipulate other organisms through DNA modification all the time.
Exactly. And in fact, the CRISPR-Cas9, that's also a natural system that was discovered in bacteria. It's a system that bacteria use to protect themselves from viruses. And so it was discovered and it was altered for this new use, which is to edit genes in any organism. So these are all natural systems that were taken and kind of changed so that we could use them in a desirable way. Let's move on to the slide that you're looking at now on the screen. And this is really just a slide to sort of to illustrate what's happening during the process of plant breeding. In the upper part of the slide, you see a curve, a normal curve. It's labeled germplasm or genetic raw material. And this represents the range of any given phenotype, which is how things look in nature. And so, for example, in humans, we see short humans, we see tall humans. Most people are somewhere around the average. And so that's a normal curve. What a plant breeder would do is to come in and by using altered selection and mating to change the mean of that population so that it's different than the original source. There are two things going on. First, the plant breeder changes the mean in a desirable way, if you want taller or shorter, for example, in humans. And it also, the plant breeder reduces the variation within the crop, within the population, so that the plants are more uniform. So you see that not only is the curve as the mean change, but also it's a tighter normal curve. So those are the two things that essentially happen during the process of plant breeding. And in plant breeding, you're always dealing with populations. You do deal with individuals, but the product that you're actually releasing is a population of individuals. So what is plant breeding? Well, I can boil it down to this. So there's a kind of a preamble in the top of the slide. You see that breeding objectives. You're not going to get anywhere if you don't know where you're driving, right? You have to know what your destination is, and so that's what breeding objectives are. It's the destination that tells you where you're trying to what constitutes success. Germplasm is kind of like the raw materials for what you're going to the genetic raw materials for what you want to make. So if you want to make a house, you have to go out and get lumber and fasteners and a lot of different things to build a house. Well, that's the germplasm is what a plant breeder uses to build a new variety, a new cultivar. And the way they do it is to take the germplasm and subject it to an altered, some kind of a strategy that includes altered controlled mating and selection. And this kind of is a cyclic process that goes back and forth. And over time, this will change the mean of the population, and also it will change the variance. And when we're done, if the finished product goes into testing and seed production, the testing is versus the standard cultivars, and if the new variety test variety performs well, it'll be advanced on and be released. The plant breeder actually only does the top part. Works to get the breeding objectives together with clients, acquires the germplasm, and then designs the strategy of controlled mating and selection, and actually executes that strategy. When the variety is finished, at least in the private sector, it's the job of a whole separate team of individuals to evaluate that new cultivar and to produce more propagules or seeds from that cultivar, and then to release and sell it, because that would be kind of a conflict of interest for the plant breeder. Plant breeder is not at that point an objective observer, since they have actually bred the material, and so they're no longer objective. So that's the role. That's what the plant breeder does, and believe me, that's plenty to do. That'll keep you very busy. Okay, so let's charge on into the different crops. This is what asparagus looks like in the field. For those of you who have never actually seen the crop in the field up close and personal, and it's just winding up now. Actually, the asparagus season in New Jersey. And so that's the marketable part that you're seeing right there. Those spheres that come out of the ground in the spring are the things that are the marketable product that's harvested, bunched, washed, cooled, and then shipped out to the outlets, either restaurants or supermarket. Where is it grown? Well, predominantly over the last 15 or 20 years, China has come on the scene, and in terms of hectares, I think they're up closer to about 200,000 hectares in 2020, which is a lot of asparagus. It's a relatively minor crop. And Peru has also increased their acreage, and northern Chile, which you don't see on this chart, is also increased. The more traditional places where asparagus was grown in the 20th century, the U.S., Northern Europe, other places in Europe and Eastern Asia, have actually declined. And the reasons for this will be explained in some of the upcoming slides. So trends, there's a couple of trends going on in asparagus. First of all, asparagus was considered a minor crop, and almost all the breeding until about 20 years ago took place in the public sector. That is, at universities and with the USDA and organizations like that. But there's been a recent shift as the asparagus seeds have grown in value and the acreage has shot up. There's a recent shift to the private sector. Private seed companies now are assuming the burden for doing the asparagus breeding. Another shift that's going on is that asparagus was produced primarily in the temperate regions of the world, in Europe and North America, Canada, but the labor costs, it's a very labor-intensive crop, and this has driven a shift in production from the temperate regions to tropical and subtropical regions like Latin America and China. And this shift to the tropical subtropics has also extended the production season, has resulted in higher yields, but it's all potentiated by things like better packing and transportation and energy costs. If energy costs suddenly shot up, or we had trade issues with China and Latin America, this could be a problem to have a continuous flow of asparagus year-round. So this is what the crop looks like if you don't harvest the spears. It continues to grow into what's called a fern stage. This is the mature foliage that photosynthesizes and produces the sugars that are stored then in the crown that'll be used to produce as energy for the next year's crop. And you can see this one is under drip irrigation, an experiment that we did at the station in Bridgeton. So here's the structure of kind of a cross-section of an asparagus plant. So it's a perennial plant where the crown, which is sort of right in the middle there, where it says crown and storage roots, is the persistent part of the plant that is always there. The shoots, spears, the mature foliage is ephemeral. It is seasonal, so it's there during the growing season, and then it disappears during the winter months or the dormant months. And dormancy can also come in the tropics, by the way. So there's the crown, and then there's feeder roots that can go quite deep into the field. And the harvestable part of the crop then is the shoots that come out of the crown, come through the soil, and as they're young and very tender, they're harvested and consumed directly. All right, so a little bit about asparagus. It's an unusual vegetable. It is diploid. That means there's two copies of every gene, and that's music to the ears of plant breeders because many plants are polyploid. They have multiple copies of genes, and that creates kind of a nightmare scenario where it's difficult to sort out the genetics. The other thing about asparagus, it's dioecious. That means that it's like humans. There are male and female plants. There are a few other plants like that, like holly and hemp are other examples. It's also a monocot, which is a little unusual for vegetables. There are a few other ones. Onions are also monocots. Perennial, as we said. Now, in the genus Asparagus includes a lot of different species, but they have not. It's been difficult to make crosses among the other species that are in that genus, unlike tomato, which we'll talk about a little bit later. So that has not been a source of any genes. No interspecific hybrids have led to anything commercial or any genes that have been transferred from wild species into the cultivated species, Asparagus officinalis. So there's 150 species, a lot of species, and almost none of them can be crossed with the cultivated asparagus. Center of origin is Eastern Mediterranean. We'll have a map here in a second. And the only one that is of commercial interest for food species is officinalis. There is I think Acutafolius is interesting as an ornamental, but other than that, it's just officinalis. All right, so here's the center of origin for Asparagus officinalis over in the Eastern Mediterranean. It is found along the perimeter of the Mediterranean Sea in in Turkey, Asia Minor, and around through the Sinai into Egypt. So if you're an asparagus breeder, you can fly into Istanbul and take a long hike around the Eastern Mediterranean, try to find wild asparagus to look for genes in all the wild asparagus populations that will impart some benefit to the cultivated crop. That's the raw materials for improvement. That's where to find them. All right, so asparagus has been around for quite a while. There are actual reliefs, Egyptian reliefs, that show people growing and consuming asparagus in Egypt as early as 2000 BC. And of course, the plant breeders were the farmers in those days, and farmers sort of inadvertently do plant breeding just because they go out into the fields, and when they are selecting seeds for the next year's crop, they always select the best individuals. So, and this was elevated during the 1800s in Europe, where finally there was organized programs for selecting vigorous females of asparagus, and this led to some improvements, early improvements of the crop. In the early 20th century, it was discovered that you could clonally propagate with the crown. You could separate the crown and cut it into pieces just with a knife. And so you could take one crown and make it into 10 plants. And that was a boon to the plant breeding. In the mid-20th century, sib mating was introduced to introduce genetic variability into breeding lines, both male and female. The big breakthrough that's more recent is during the 1980s, the AllMale hybrids, which we'll talk about in just a minute. And the two main ones that were the early ones that came out, UC 157 out of UC Davis, and Jersey Giant, which came out also with Howard Ellison during the 1980s. And this revolutionized the whole asparagus breeding industry. Since then, the whole range of germplasm has been expanded, and really asparagus breeding is a very hot area right now, because asparagus is viewed as being kind of a designer crop that is highly nutritious, and actually it is. It does have high nutrition. All right, so how does a breeder figure out what to do? How to improve the crop? Well, it depends on who you talk to. If you talk to the farmers, the producers, you get one answer. They want yield, concentrated early harvest, extended season, disease resistance. Whereas if you're talking to a processor, they want uniformity of the spears for pre-cutting and packaging, consistent spear girth, extended shelf life so it can be shipped long distances. The consumers don't care about any of that stuff. They just want a nice tender spear that has good flavor and has high nutrition and convenience. And so the plant breeder has to figure out how to balance these different clientele groups to develop kind of a composite cultivar that will please everybody. Because if you don't please everybody, that's kind of a broken link in the chain, and you're going to get nowhere. All right, back to this issue of being dioecious, which means that we have separate male and female plants. These are the flowers, what they look like. So the top one, you see staminate, which is the male flower. So the male plant has flowers that look like that. And then the lower left is pistillate. That's a female flower. Very uncommonly, there are hermaphrodites that form, and it turns out those are important, even though they're uncommon. And they're very important to the plant breeder of asparagus, and we'll talk about those more in just a second. You see those letters that follow the staminate, the pistillate, and hermaphrodite, big M, big M, big M, little m, little m, little m after the pistillate. We'll talk about that in just a second. The M is actually a gene that controls the sex. Much like in humans, the XY chromosome is a male and an XX is a female. It's more or less the same in asparagus. So in a normal regular population, big M, little m would be a male, and little m would be a female. Okay, so you can see just from this picture what those flowers look like in reality. So they're quite small, but they're loaded with nectar, and so insects, they're pollinated by insects, and bees and flies just really are effective pollinators for this crop. And it's obviously cross-pollinated, because if you have separate female and male plants, they must be cross-pollinated. Otherwise, there would be no fruit set or seed set. In the case of asparagus, it turns out, unlike most places in nature, that males are more desirable than females. It's males, it turns out, have the male plants, if just planted as a population, have higher yield. They usually start production of spears earlier in the spring than females. Of course, males do not produce fruits. Those fruits are very energy intensive, and it diverts nutrients from crowns and storage roots that can contribute to the vegetative health of the plant and the yield of spears the following year. Males are less likely to lodge in storms and strong winds, because the load of berries weighs the plants down in females. There's no volunteer seedlings in fields of males, since there's no fruits that drop to the ground and the seeds then germinate. And they tend to live longer in the field, 15 to 20 years, as opposed to maybe 10 to 15 years. So there's a lot of advantages of males over female plants in asparagus. Now, this is partly because we're harvesting a vegetative part. We're harvesting a spear, a shoot. And so in most crops where you're harvesting a fruit or seeds, you really have to have that reproductive function. You have to have the function of the female for the crop to be successful, but that's not the case in asparagus. Sure.
There's a lot of, I guess, because of the seeds with some of the female plants, there's quite a few wild asparagus plants growing around. The birds distribute them where you do have female plants. So you probably see more of that in South Jersey than anywhere else, I would imagine.
Oh, absolutely, yeah. The berries, the asparagus berries, are not very palatable to humans, but birds love them. And as they mature, they turn a bright crimson, and the birds come in August when the berries are maturing, and they eat them and just spread them all over the place. And so we've got wild asparagus growing in the hedgerows and the ditches all over in South Jersey. It's a very, very common weed as a consequence. Even though, a lot of the crop now is grown to these all-male hybrids, which don't have any females, or very few females, some of the wild asparagus grows up, produces enough berries that it sustains itself as an escaped weed. So, the berries are very effective as far as distributing asparagus as an introduced weed. All right, so I'm on to the next slide here, and to talk about this M locus that controls the sexes in asparagus. So as you saw from the previous slide, little m is female, big M, little m is male, or big M. We'll talk about that in just a second. Now, in a normal population, in a population in nature, you'd have half males and half females, just like you do in humans. And when males and females mate in humans, on average, half the offspring are male and half are female. And that's the same case that we have in asparagus. Okay, so that's a normal wild asparagus population would have half males, half females, and when they mate, they produce half males and half females. It's possible to develop what's called a super male. Now, this is, in this age of superheroes, it's something that all of us would like to be, is a super male. But sadly, only asparagus can be super males, and this is how it happens. We start with known male plants, and we know that because they don't produce any berries, and they have this genotype in a regular population, big M, little m. So that's a male. All right. So on rare occasions, as I said, hermaphrodites are rare, and the flowers are small. They'd be difficult to see. The only way to find a hermaphrodite is to look for a fruit set or a berry that occurs on a male plant. The only way for that to happen is for a hermaphrodite flower to occur, or a female flower, but that's even more rare. So when you see a mature berry on a male plant, it means that you had a hermaphrodite, and that hermaphrodite was likely a self-pollination event from the plant. from that plant that the berry is on. All right, so we've got a hermaphrodite flower. What's next? All right, so that's a self-pollination, and in effect, it's a cross of the two heterozygotes, big M, little m. If we do that, 25% of the progeny of that cross will be big M, which is called a supermale. It does not occur in nature. Just like we don't see YY males in humans, and in fact, in humans, it would probably be lethal. But they're fine in asparagus, and they don't look any different. But we call them supermales because if you cross them with females, all the progeny will be male. Okay, we'll see that result in the next slide. The other feature of asparagus breeding programs in the 21st century is tissue culture. And a lot of that technology was developed right at Rutgers. So the development of specific growth regulators that made it possible to micropropagate very efficiently asparagus. And so it's used to propagate both the male and the female parents in the breeding program. All right, so here's the actual process of breeding all-male hybrid cultivars of asparagus. We start with germplasm, that's the source population, and from that we extract the beginnings of what will be the female, which is the little m on the left, and the male parent on the right, the big M, little m. And then we start to breed those separately as populations. All right, so there's a whole breeding process. We won't get into a lot of the details to develop a inbred parent that could be used to develop a hybrid variety. And so we're going to end up with a female at the end of this breeding process and a male. And the male is going to be a super male. So we are breeding this male, this big M, little m, selecting it for all these features so that when it becomes an inbred, the resulting hybrid will have high performance. The next thing the breeder does is to look for a berry on the males, the hermaphrodite flower, and then to self-pollinate it. And when you do that, 25% of the progeny, as we saw on a couple slides ago, will be super males, the 25% big M. Those are super males. All right, so we micropropagate both the female and that super male. When you micropropagate, you're clonally propagating. It doesn't make any genetic changes. And so you're taking one or a couple little m's, and you're producing thousands of plants. And the reason is to get large enough populations to conduct seed production. And so we end up with large populations of little m, the female parent, and big M, the super male inbred parent. And then finally, those are interplanted in a field, and pollinators are added to take the pollen from the super male parent to the little m female to produce large quantities of all male hybrid seed. All the crosses from little m and big M will have the same genotype. It'll be big M, little m, and that's a male. So 100% of the progeny of that cross will be male, which is what we want because the males in asparagus are superior to the females. So this is a modern asparagus breeding program, how it operates. And part of this was a lot of this technology was actually invented at Rutgers. All right, so what is the advantage of these all-male hybrids compared to the old-time cultivars? Well, you can see here from this slide, Mary Washington was an early 20th century cultivar, still around actually. You can buy crowns of Mary Washington, from the major catalog houses. The yields, and that Mary Washington, when you plant it, is about half male and half female plants. And they both produce spears, but the females produce many fewer spears than the male. So it's only 1,000 pounds per acre for Mary Washington. Both Jersey Giant and UC 157 produce about 4,000 pounds per acre. Jersey Giant is more adapted to cooler regions, and UC 157 more to warmer, to subtropical regions. There are value-added traits in asparagus. There aren't too many of them, but one of them that I like is this purple asparagus, which is simply anthocyanin. It's a health-related compound that turns the surface of the spears purple. And for some reason, the cultivars that have been developed have advantages to green asparagus. They have larger spears. They have less fiber in the spears, higher sugar levels, less saponin. The saponin has health benefits, but it also has imparts poor flavor, and usually actually higher sugar in these purple skin varieties. So if you can find these things on the market, they're just beginning to appear now. I would urge you to give them a try. I think they're really excellent.
So, is there something linked with the color and the different characteristics per se, directly linked?
Well, it turns out a lot of these purple cultivars are tetraploids. So they have 4 copies of all genes instead of 2. And for this reason, that probably accounts for the larger spears and maybe the higher sugar, sweeter taste. But it doesn't probably explain everything as to why the purple asparagus is so much different than green. But it's a big part of it.
Thank you.
All right, so how do we make asparagus crosses? We did a little pictorial with the asparagus breeder, Dr. Stephen Garrison, and this is your tool chest right here. And this is kind of typical for a plant breeder. You're constantly inventing new little gadgets and devices. And so this is what Steve invented. He's got a little block there with a toothpick and a little hole for the toothpick, and he's got holes to hold the microfuge tubes, long neoprene tube, and an aspirator. We'll see how this stuff all works together to enable Steve to make crosses. Well, the first thing he does is to aspirate the pollen out of male flowers. You can see how small they are. So Steve is holding a male flower, and there's a little tiny aspirating tube that he sticks into the flower and then just sucks on it to create a vacuum, and that pulls the pollen into one of these microfuge tubes. And then he uses that toothpick, another high-tech device, to transfer the pollen from the microfuge tube onto the stigma of the female flower, which then goes on to produce a berry that contains several seeds in it. And that's a hybrid between the male and the female that were selected. And it's possible to grow asparagus in the greenhouse. It's not a greenhouse crop. You can't really produce spears in pots. You can grow them in greenhouse, if you're growing in soil. But so plant breeders will often grow plants in the greenhouse just to get them started as transplants. And as I alluded to before, the Rutgers has been a very important factor in the development of both new cultivars of asparagus and also the AllMale breeding technology. And all of these things have been exported around the world, and so pretty much all these strategies are being practiced by many different seed companies and other universities that have asparagus breeding programs. But it turns out Rutgers no longer does. The program was sold in 2011, 2012 to the Bijo seed company in the Netherlands. And the money came back to the experiment station, and a small portion of the breeding program continued. But most of these cultivars, Jersey Giant, Knight, Supreme, King, Deluxe, reverted to ownership by Bijo, and they're still available on the market. And we consider this evidence of success of the program. If you can develop a program in the public sector rather, and then the private sector comes along and says they want to purchase the program and continue breeding asparagus, in the private sector, that really is evidence of the success of the program. So this is also an example of how things are shifting with asparagus from the public sector to the private sector. But these are all cultivars that were bred right here in Jersey and still have these names and are grown around the world. All right, so that's all I had on asparagus. Does anybody have any questions about asparagus? If not, we'll proceed on to tomatoes. All right, so tomatoes are a very, very popular crop. They're, depending on how you measure them, they're either the number 2 or 3 in terms of volume or acreage. And most of the growth of the crop now, in terms of increased acreage, is taking place once again in China. And a lot of this acreage in China is processing tomatoes. And we'll talk about processing versus fresh market in just a second. The acreage in the U.S. is fairly stable, but over time it tends to decrease, whereas in places like Turkey, India, Mexico, which you don't see on here on this list, the acreage is increasing. The fresh market tomato crop is highly labor intensive, and for that reason, a lot of the fresh market crop is going, to areas like Mexico, Latin America, and certain areas of Asia. Processing tomatoes are machine harvested, and so a lot of those are still grown in the U.S. But there's a shift now to China and Mexico and to Brazil. A lot of people think that the tomato was actually is native to Italy. It is not native to Italy. And in fact, the production of tomatoes in Italy is dropping, where the world production of tomato continues to increase. All right, so some facts about tomato. It's a nightshade, so it's related to other crops like peppers, tobacco, eggplant, potatoes. These are petunias. So that's the family to which tomatoes belong. And it's actually a tropical crop that was domesticated in the higher elevations of the western flank of the Andean Mountains in South America. It grows as a perennial in the tropics. It has a woody stem, but it grows as an annual in the temperate zones and has been bred as an annual. Like asparagus, it's a diploid. It has 2 copies of every gene, but has a different chromosome number. I wanted to pay homage to Dr. Charlie Rick, who sort of single-handedly during the late 20th century made tomato into not only a very popular crop for culinary reasons, but also a scientific organism. And so tomato is a very popular organism for people to work on just to produce science, and that's in large part due to the efforts of Dr. Rick that we'll talk a little bit about in the coming slides. So autogamous, which means that's a fancy word for self-pollinating. Self-pollinating means that they will, under normal circumstances, if you take the seeds out of a fruit, it results from a pollination event from its own genetics. So the pollen and the egg from the same plant fuse, and that's self-pollination. So tomato has been bred as a self-pollinating crop and is very tolerant of self-pollinating. Most cross-pollinated crops are not tolerant of inbreeding. If you take an inbreed, for example, humans, it can have devastating effects. If you take an inbreed corn, it can have devastating effects. That's not true of tomatoes. They're very, very tolerant of inbreeding. It's very easy to manipulate tomatoes in the test tube, either molecular or cellular manipulations. And tomato was the first GMO organism in the early 1990s. Some of you in the audience might recall the Flavor Saver tomato from the early 1990s that made a lot of headlines, but didn't make much headlines in the food section. That was discontinued because number one, it was a bad product. Number two, there was such a backlash that particularly in Europe, to GMO, and it kind of forced the U.S. to take a step back and look at regulation of GMO. And while labeling of GMO in the U.S. is still not a requirement, it's still a relatively unpopular technology to apply to plants. Mainly for social and belief reasons. It really doesn't. There aren't any stigmas really associated with a GMO plant, other than it may contain a gene from another species, but there really is no scientific reason to say that it's an inferior plant. All right, so this odd-looking gentleman here is Dr. Charlie Rick, and he actually looked just like that all the time. He had an old hat that he just wore everywhere, and he was quite a character. He died in, I think, in 2005, but he was instrumental in going to South America dozens of times to collect wild tomatoes, bring them back to the U.S., organize the collection, which ultimately turned into a cache of germplasm that has propelled tomato to the position of being a model species for geneticists. So that was quite important work. And the tomatoes, as I said, are from the eastern or the western flank of the Andean Mountains along Colombia, Ecuador, Peru, and northern Chile.
Tom, I have a question about that. In reading about the origin of the tomato, you find that the origin of its relatives existed in some pretty harsh climatic conditions, from extreme heat and drought to colder temperature extremes with a lot of moisture. So within that, was that what Charlie Rick was trying to do, trying to collect all of that genetics so that he could try to bring some of that, those characteristics and some of the disease resistance back into some of these crops?
Absolutely, Bill. It's a very good point. We'll talk a little bit about that in the coming slides, but yeah, it's like not only resistance to cold and heat and drought, but also to salt, a lot of disease resistances, and a lot of these genes are actually in the cultivars that are on the market now. Came from material that was collected from the wild by Dr. Rick and then used by plant breeders all over the world to extract the genes and put them into modern cultivars. All right, so tomatoes have been domesticated into, unlike the asparagus where we have maybe purple and green types, we have several different types of tomatoes. We have the fresh market type, the big kind of soft multilocular types that you're used to in the grocery store and on the farm stands, heirlooms that are even more soft, fresh market plum tomatoes that can be probably used for fresh market or for paste, cherry tomatoes, grape tomatoes, and processing tomatoes. Well, I'll show you a picture of one, and I think it's the next slide, but it turns out that most of the acreage in the world in 2020 is processing or canning tomatoes. All right, so the processing tomatoes are on the lower right, and they tend to be kind of smaller than fresh market tomatoes, but they're very low in water content, so that when you don't have to use much energy to cook the water out of them to make sauce and paste. And so, but they're not edible as fresh market tomatoes. You can't slice them and eat them. They're not very palatable. And we have the small fruited types, of which Rutgers has been involved with the breeding of grapes and cherries. We'll talk about later, myself or Pete. And we have done some fresh market breeding, but I've actually been involved with a lot of processing breeding myself. So we're involved with tomatoes on many different fronts at Rutgers. All right, back to the wild species that are found from South America. These are all the species that are cross-compatible with cultivated tomato. There are a lot of other tomato relatives that can be found in South America, but many of those can't be successfully crossed with tomato. And so this is the source of most of the genetic variability for things like disease and cold and heat and drought tolerance have come from all these wild species that you see listed in this table. You can see they all have 24 chromosomes, which means that they're probably quite similar, but that the mating system is quite different. Cultivated tomato it's SP, which means self-pollinated. Many of these wild species are either cross-pollinated, like Kimluskii, or actually self-incompatible. They have a mechanism that prevents self-pollination. So most of the wild species are actually cross-pollinated. Some are self-pollinated, but many are cross-pollinated. So it's likely that during domestication that tomatoes were converted from being crossed to being self-pollinated.
I'm sorry, do we know which one of these was actually the origin?
Probably it was Pimpinellifolium.
Okay.
Pimpinellifolium it can be found still in the wild. The current tomato, you can see it up on this slide here. The upper left is and tomato is technically botanically a berry, and you can see why in this picture. It definitely appears to be a berry. And see, here's the fruits of some other wild species, Cheesemanii, hirsutum, that look sort of similar to tomato, but quite different. So they can be found in nature to have many features of cultivated tomato that are similar, but they're definitely adapted for the wild. That Cheesemanii actually grows on the Galapagos Islands off the coast of Ecuador, and it's a food source for the giant turtles that live there. All right. So the other thing that Charlie Rick did was to accumulate mutants. So plants that look different, and then when you did the genetic studies, you found out that the difference, the plants look different because they had a mutant gene. And he accumulated about 3,500 of these different mutations during his career. And you can see the little pictures of kind of what these mutants look like on the right, but there were just thousands of them. And some of these mutants were used to unravel, biochemistry. For example, this pathway that you see here is how carotenoid pigments are made. It starts, at the top with mevalonic acid, and then when we get to the bottom, we see things like beta carotene, like delta carotene, lycopene, neurosporine. These are all carotenoid pigments that cause the fruit of the tomato to look the way it does. On the upper right there, you see the red, orange, and yellow tomatoes. Well, that's due to the different accumulation of these pigments. And by employing these mutants, it was possible for physiologists to unravel this pathway. I'll just point your attention to one of these mutants. Is kind of right in the middle of the slide. It's something called OGC, and it's called crimson. And this is a fairly popular mutation for plant breeders because it blocks the formation of beta carotene in the pathway, and it promotes the formation of lycopene, which makes the fruits more intensely red. And lycopene is actually, of course, beta carotene is provitamin A, but lycopene is also a health-related compound. It's an antioxidant. All right, now this looks busy and kind of complicated. It's actually a map of the 12 chromosomes of tomato as of about 32 years ago. We don't even, we have so many genes on this map now, it's impossible to actually put it on one page. But you can see, for example, so all the genes of tomato are located on one of these 12 chromosomes. And on chromosome 6, you can see kind of in the middle, the blue arrow there to the circle, that is the MI locus there is a nematode resistance locus. And the red circle and the red arrow is where that OG gene is located. So the geneticists know how to use genetics to find the location of these genes on the chromosomes. And once we know where they are, we can target them for improvement. As of 2011, almost 10 years ago now, the entire genome of tomato was sequenced. Hundreds of millions of bases, starting from one end of chromosome 1 to the other end of chromosome 12. So we know every single base that's in the genome of tomato. Many different crops have been sequenced, and in fact, in 2020, it's relatively easy and cheap to do that. But this is a huge advantage because we can find any gene if we know what its sequence is in the genome of tomato. All right, so in this slide, if you're a tomato breeder and if you're breeding for disease resistance, you no longer go out in the field and look for the disease. What you look for is a gene, is a small DNA sequence that's linked to the disease resistance gene. This means that you don't have to mess around with the field, you don't have to mess around with the pathogen, you don't have to wait for the disease to form. So this is a situation where we have determined the genotype of all these different inbreds by using SNPs, or single nucleotide polymorphisms, to look for disease resistance. So in this table, S is sensitive, R is resistant. And, so this can be done very quickly within a couple of days at a very low cost. And this is what's called marker-assisted breeding. So we can breed for the markers that are linked to the important genes rather than going out and doing all the work in the field to breed for the genes themselves, which are a lot more expensive and a lot less effective. All right, so we're going to talk a little bit about the breeding at Rutgers. Tomatoes, why breed tomatoes in New Jersey? Well, this used to be the garden state. Well, it is the garden state still, but it used to be really the tomato capital of the country as well. But over time, that has changed, and a lot of crops, including tomatoes, have shifted their focus to other parts of the country, to the south, where there's a longer growing season, and also to the west, to California and Florida. There are other places around the country that grow more tomatoes than New Jersey. We're down probably less than 3,500 acres of tomatoes in New Jersey in 2020. At one time, we were up closer to 40,000 acres. So it's substantially less than what we. But those 3,500 acres do not have access to adapted cultivars because they are all bred for the conditions that are prevalent in California and Florida. And when you put those varieties into New Jersey, they don't necessarily perform well. They may perform well, and in fact, many varieties that are bred in either California or Florida do perform well in New Jersey, but it's not a guarantee. We also have value-added product niches in this whole, all these big metropolitan areas, and you know, some of the higher-end restaurants and things like that. So we have these other opportunities, you know, that are more niche kind of markets that are available to us, but we need to have specialty tomatoes to fit those niches. All right, so the breeding programs that we have here at Rutgers on tomatoes are sort of a balance of farm production, disease resistance, yield, fruit quality, maturity, with consumer kind of attributes that are considered to be more value added, things like flavor, visual appeal, nutritional value, or novelty. You can't have one without the other. You can't have a value added fruit without having, you know, some measure of yield, you know, holding ability in the field, disease resistance, and that sort of thing. And in New Jersey, at least, you're not going to compete with California and Florida for commodity grade fruits. It's just there's no way to beat their costs. So we're kind of stuck in New Jersey where we need value added products, but we need also for those products to perform in the field. All right, we'll talk a little bit about tomato breeding. It's a little bit different than asparagus. Most tomatoes are F1 hybrid cultivars, both processing and fresh market. But this is a business strategy by the seed companies. Cultivars of tomato, they don't need to be hybrid. They can perform just fine as just what's called OPs, or open pollinated populations. But the seed companies make a lot more money on the hybrids, and so they have kind of marketed the whole strategy of hybrid cultivars and tomatoes, and they get a lot of money for the seeds as a consequence. So we start with the germplasm, just like a breeder always does, and we pull out females that are incipient, the ones that tend to have higher seed yields, and males that tend to have higher pollen production. And we're going to produce two different parents, two different inbreds, a female and a male. And so the plant breeder, along the way, inbreeds and also selects for the best plants in both of those different populations, so that we end up with two parents that, when we cross them together, are going to produce a hybrid that has high performance. So we can produce either an F1 hybrid between those two parents, that's a cross between the female or the male, or we could just release an OP, which is the female parent just crossed with itself. If we do that, the seed company doesn't make much money, but they make $2,500 per pound on F1 hybrid seeds. Now, there's no easy way to produce hybrid tomato seeds. It has to be done by hand. And so you can guess where all that takes place. It's not in the U.S. It's done overseas, predominantly in Asia. So it is rather expensive and unwieldy to produce F1 hybrid tomato seed, and in my view, it's not necessary. But the seed company, you know, when there's money to be made, they'll go ahead and do this. You know, and you know, shipping all the inbreds over to Asia and then shipping the hybrid seeds back, it's really quite quite tedious and expensive. But that's the way the industry works. All the cultivars that have been bred at Rutgers by myself and Pete Nietzsche are OPs. We think that that's probably more defensible for a public university to be breeding cultivars that are OPs than hybrids. The cultivar Ramapo that was released in 1968 was an F1 hybrid, is an F1 hybrid. So that's all the other cultivars released by Rutgers are OPs. Does everybody know the difference between an OP and a hybrid based on, for example, this slide? Well, an F1 hybrid is a cross, the seed from a cross of two different individuals. In this case, you can see we're crossing a female inbred parent with a male inbred parent, so it has two parents, like a human would. Whereas an OP is you're just taking one population and allowing it to interbreed randomly among the individuals within the population, and so the pollination is not controlled. And for that reason, it's called an OP, or an open pollinated population. The F1 hybrid, the theory is that it has higher performance because it has hybrid vigor, or heterosis. In tomatoes and many other crops, there's also the advantage that you can combine disease resistance genes from the two inbreds. So if you have a female that's resistant to three pathogens and a male that's resistant to three different pathogens, the F1 hybrid will be resistant to 6 pathogens. So it's additive. So there is an advantage as far as disease resistance to these hybrids, but as far as the overall performance, I think it's the value of the hybrids is not proportional to the cost of producing them in tomatoes because they're self-pollinated.
Some people are confused about the traditional breeding you're describing versus using other types of transgenic breeding, because of the way they're asking questions, it appears that they're asking whether or not you can revert these species back to their original form, and can you explain how this traditional breeding differs from, say, like, using transgenic methods?
In the traditional breeding, we're only using whole plants, and we're using, genes that are from naturally occurring within the species. We can also use, I think, for an example of tomatoes, crosses with different wild species. So those are all considered to be traditional breeding. Those are things that have been done for centuries, where we alter the mating, we cross two individuals, and then we simply select in the population for the individuals that have the highest performance. Non-traditional breeding would be to take a gene from another species that's not cross-compatible with tomato and moving it into the crop using a vector like Agrobacterium. So I would consider that to be not to be consistent with traditional plant breeding. And in fact, it has the label as genetically modified organism to designate the fact that it's and in fact, it is changed. A GMO organism has a little footprint from the Agrobacterium TI plasmid that's in it, and that's a permanent genetic change from a bacterium. So, whether this has any effect on the quality of the fruit is still a matter of debate. But so I would say that the difference between traditional and non-traditional is that traditional is all done in whole plants in the field and only by sexual hybridization of plants to move genes from one plant to another. Whereas non-traditional, it takes place in the laboratory, using vectors, using isolated genes, or some other non-sexual way to induce genetic variability.
Also, another thing, we have some people asking, like with specific traits, if you wanted to have low acid in tomatoes, how would you go about like with specific characteristics as a breeder, how do you approach that if you wanted to develop a lower acid tomato or, a specific characteristic? How would you approach that as your trial, your goal?
The traditional approach would be to evaluate germplasm and look for a population or an individual that has fruit that has a low fruit acidity, and then to do the genetics to determine that, how is that low acidity, what's the inheritance of it? Is it a single gene that causes that, or is it two genes? And then once you determine that, to design a breeding program to actually move the genes that cause low humidity or low acidity rather in the germplasm plant into the cultivar. And there are many different breeding approaches that you can do that. The most popular would be a back cross. The non-traditional approach would be to actually go into the genetic apparatus of the plant, find the gene that controls fruit acidity, and to alter it. Like, for example, with CRISPR or something, to go in and actually change the gene so it makes a protein that alters the acidity. So that's an example of the traditional versus non-traditional approaches to breed for low acidity fruit and tomato.
Another question somewhat related. The person had here, for tomatoes and asparagus, how many back crosses does it take for a new characteristic to be stably expressed in a new cultivar? How much does the number of back crosses to establish a new cultivar vary among breeding methods?
Not that much. It turns out that back cross, probably 3 or 4 back crosses is enough to recover over 95% of the genome of the recurrent parent. That's the cultivar that you're trying to back cross the trait into. So about 3 or 4 would be a rule of thumb. Sometimes it takes more back crosses, particularly if you have linkages that are undesirable that need to be broken between genes. You have an undesirable gene that's linked with a desirable one. It might take more back crosses to break that linkage. But typically, it wouldn't really matter that much between asparagus or tomato or any other crop. Probably 3 or 4 back crosses should be enough.
Thank you.
Okay. All right, so we get two generations per year with tomato. And that's not always real easy. There's some late ones that are difficult to squeeze into this. And the summertime is short. What's going on right now, as you see, we're just starting. And usually we just evaluate cultivars during the summer. And then the winter is, we do predominantly in the greenhouse. It's a much longer season, and that's where we grow small populations in the greenhouse and make all the crosses. So we can get 2 generations per year. So this helps to kind of move things along. In New Jersey, we have 3 main locations where we either test or breed tomatoes. I'm actually stationed in the South vegetable station, the Rarick, which is near Bridgeton, New Jersey, in Cumberland County. And then we evaluate a lot of the breeding lines at the Snyder Farm up in Hunterdon County. We do some seed production and performance trials at Port Farm 3, that right now is near campus in New Brunswick, or East Brunswick. But that facility is currently shut down while they're fixing the irrigation system. So this gives us a broad range of soil types and climates that will help us to gauge, how varieties perform under different circumstances and different environments. We also select for composite characteristics. As I said before, it's a balance between many different things that differentiate a good from a great tomato. And a great tomato has characteristics that are desirable in many different categories. So here's we see earliness, yield, disease resistance, external color, internal color, bricks, and there are many more characteristics that we evaluate, and we try to get a score for those, and then select for the breeding lines that tend to have not just one high score, but high scores in all those characteristics. We're going to talk a little bit about flavor. And here's a tomato fruit. I think there's a picture actually taken by Pete Nietzsche, our photographer. And tomatoes are about 95% water. You know, a lot of vegetables are mostly water. And you can see that of the other components of tomato that make up the tomato, there's only really 3 that affect flavor very much: the sugars, organic acids, and volatiles. And of those, they only make up about 3% of the tomato. So about 100% of the tomato flavor is determined by 3% of its constituents, of its chemicals. All right, so closer look, you know, here's all the sugars. The main sugars, fructose, glucose, sucrose, are the main contributors to sweetness. The tartness are organic and amino acids, and there's about 3 or 4 of those that are important. And so we breed typically by looking for those or measurements that estimate those. The volatiles are relatively more difficult to breed for. There are over 400 of them have been characterized in tomato, and there's only about probably 25 of those that really have any effect on flavor. But they can have a huge effect on flavor, the volatiles. And breeding for flavor, if you just rely on humans to tell you what tastes good, you'll get nowhere, because the lady on the left will tell you that tomato that she's just tasted was lousy. The guy right behind her will say that's the best tomato that ever was. And this is characteristic of almost every tomato tasting event I've ever been to. You know, maybe Pete has a different experience with all the tastings they've had at Snyder Farm, but you know, there's a lot of variability in these readings, and so it's almost impossible to use these data as a guide. Although it has to be done, we ultimately, you know, people have to taste tomatoes to say whether it's good or bad. But by relying on that for breeding, it's very, very dubious and difficult.
When I've tasted tomatoes at the different sites, the same exact variety will taste totally different depending on soil types and practices growing the tomatoes. So there's a huge variability even within that. Even when I've gone to, you know, there was quite a few years ago when we were going to the taste tests in South and in North Jersey, and I noticed huge differences in the flavor of the same exact tomatoes. So I know soils and environment and fertilizers used and all that play a big role in that.
You're absolutely 100% correct there, Bill. It's like, and that makes it even more difficult to breed, because what are you breeding for? Which location are you breeding for? What tastes good in one location, you know, may not necessarily taste good somewhere else. And so this is a huge challenge. And so we're looking for things that, you know, have more or less universal good flavor. I think it's probably safe to say that organic acids and sugars are relatively stable. I think it's more these volatiles that change as you change the environment and the soil type. And so for that reason, we've tended to focus more on sugars and acids than we have on volatiles. The volatiles are also very difficult technically to measure and to characterize. So as a breeding tool, you know, you can tell that I'm not very smart. So I need something that's simple. So it's like the sugars and the acids are relatively simple to measure, and as a breeding tool, they work quite well. So that's our strategy here, and I think it's effective to breed tomatoes that are relatively stable across environments. All right, so this is an example of how one volatile called foraniol can be different in tomatoes. This is on a per weight basis. So you see that in this panel of different varieties that the per weight basis of foraniol, parts per billion, varies from 24 all the way up to 418. So it's a huge range. And in general, on the far right, we see a flavor assessment column from these same cultivars that were evaluated in human taste tests. And you can see a fairly strong association of the foraniol parts per billion scores with flavor assessment of these different cultivars. Should note that Tasty Lee is a Florida variety that was selected for flavor, and you can see that it tastes good in Florida, but does not taste good in New Jersey. All right, we'll talk a little bit about acids. We're running out of time, so I think we ought to just kind of truck forward here. We see genetic variability in acids. We talked a little bit earlier about low acidity, but in this particular set of populations, we can do tests to show that there's probably 2 genes involved in the determination of fruit acidity in tomatoes. So if there's 2 genes, it's relatively easy to breed for it. The more genes that are involved, the more difficult it becomes. And here's an example of some of the different volatiles of the 25 that have some effect on flavor, and you see some notion of what the concentration is and the sorts of odor characteristics or flavor characteristics that they impart to tomatoes. So that's just an example of what volatiles can do to tomatoes. Essentially, they're what make tomatoes taste like a tomato instead of tasting like a peach. All right, well, this is flavor is positively correlated with acidity in our studies, negatively correlated with pH, which is the same measure in effect, positively correlated with total soluble solids, which is to say the sweeter the better. So the more sweeter, the higher the sweetness and the higher the tartness, at least among the tasters in New Jersey, that's what's preferred. And we found that other factors, fructose, glutamate, foraniol, geraniol, other volatiles can have strong effects on tomato flavor. And so that's what we kind of use as our basis for doing selections for flavor in tomato. We're going to talk, I'm going to skip over Rutgers and talk a little bit about the 2 cultivars that we released, and then we'll finish up. So we started breeding, Pete Nietzsche and myself, these populations that eventually became Rutgers 250, and we started doing this in 2011 with Campbell's Soup. And this is a traditional breeding approach. It's called a pedigree breeding approach, where we cross 2 different things that we happen, in this case, to get from Campbell, and then we inbreed after we've made the hybrid and select as we inbreed to the point where we reach about the F8 at the bottom there. And by the time we reach the F8, the resulting cultivars in a self-pollinated crop will be fixed or homozygous. In other words, all the plants look the same in the population. So by 2015, we were finished with this breeding program, and we went through an intensive evaluation during that year that was grant funded that resulted in the selection of Rutgers 250 and then release in 2016. And maybe many of you have acquired this cultivar and tested it out. We're always happy to hear whatever feedback we get, but we're quite happy with this variety that was targeted for gardeners. It's an open pollinated cultivar. All right, so this is the seed packet that you can get from New Jersey's Ag Experiment Station if you go on our website. And this is a copy of the sheet that was the little paper that was distributed when we released it in 2016 at the Vegetable Growers Association meeting in Atlantic City. It looks a lot like the original that was for the Rutgers that was released in 1934. So that was kind of our play on the symmetry between the 2 releases. And in 2016 was the anniversary of Rutgers University, the 250th anniversary. And so this is kind of like part of that celebration. And here's a flat of fruits of Rutgers 250 that show one of the prominent features, which is this kind of navel blossom end, the indentation there, that growers don't like because there can be rot that can form at those cavities. But most people don't seem to be too alarmed by it. All right, so then the latest one that Pete and I have been working on since about 2013 is a grape cultivar, which is bicolor, and it's a unique bicolor, sort of yellow and pink. And we just finished a plant variety protection, sort of a patent application for this cultivar, and it's been released this winter as under the name Scarlet Sunrise. And so this seeds of this cultivar are available, and I'm sure are, despite the pandemic and problems with distribution there, I'm sure it's going to be grown quite widely in New Jersey this summer.
Tom, yeah, I just wanted to say that I had tried that tomato when it came out. I know Peter brought some to one of our meetings. I thought the flavor on that was outstanding. I just wanted to let you know it was very good.
Thank you very much, Bill. I appreciate that. And I'm sure Pete appreciates that too. He being the more tomato connoisseur than I am. But we got the flavor actually from this red cultivar, SEM 9137, Seminus. And about 10, 12 years ago, we identified this cultivar as being a flavor standard among all tomatoes. And Pete also found this interesting Isis Candy cherry tomato cultivar that's like kind of a soft, bland, low yield, crack susceptible heirloom type. And we liked the color of it though. We didn't like much else about it. And so we made that cross, and ultimately we're able to combine the flavor and the firmness from 9137 with that color characteristic from Isis Candy in Scarlet Sunrise, which you kind of see in the middle, lower middle panel there. That was the original name of it, TG25.1C, renamed Scarlet Sunrise. All right, so you can see all kinds of different grape tomatoes here, and Scarlet Sunrise sort of at the right side there with a box around it. And we're thinking about another one that's sort of a sister line that has a little bit darker color, and I'm portraying it. I'm kind of walking out on a limb here saying the Scarlet Sunset. We may or may not be releasing this in the future. We'll see. We're still evaluating it, but you know, kind of a yin and yang of Scarlet Sunrise and Sunset. I'll still have to talk about this and with Pete as we go forward. But these are indeterminate. They're mid-late maturity. The fruits are very firm, almost as firm as 9137, resistant to cracking under moisture stress. And as Bill mentioned, the flavor is really quite excellent. And when we test this, the final technical set of slides here. So here's the 12 chromosomes of tomato. And so in Scarlet Sunrise, you know, sort of half of those would come from 9137 and half from Isis Candy. And we actually did the genetic test, the marker test, to show roughly half the genetic of the genome is from Isis Candy and half is from 9137. So the genes for the yellow parts of the genome here are, there's a gene somewhere in there for the color, and the flavor and firmness are somewhere in the red parts of the genome. So this is kind of using markers and to select for characteristics that have horticultural importance. So that's, I think we'll just finish here. So this is kind of where we're going. We've released Rutgers 250, Pete and I, and Scarlet Sunrise, and we'll be going down the road and seeing what else we can do here. But you know, I'm interested in maybe this next population that may or may not be Scarlet Sunset. We'll see. So that's kind of in a nutshell. I've taken a lot of elements here and tried to cram them into a presentation here that originally also included some notion of how to be a backyard plant breeder. And we won't have time to cover that very much. I have a whole separate talk for that. But really, plant breeders used to be farmers. There's nothing that prevents anybody from going in their own gardens and doing some plant breeding. It just takes, you know, finding the plants that look good, crossing them, and selecting them over and over again until you find something that's unique and better than what you started with.
Tom, what would be a good plant for somebody to get started with if they were going to do crosses?
Well, I think peppers and tomatoes and cucumbers there would be good, or squash, you know, zucchini. Those would be good vegetables to start with. I wouldn't start with things like strawberries or brambles. You know, things like that are there's those. For example, strawberries are polyploid, and there are many different ornamental species. You know that things like gerberas and things like that. that there are probably undiscovered forms of flowers that, and I know that most people in the audience probably have strolled through their garden and seen something unusual, and in many cases there's a genetic basis for that could be seized upon and could be the basis for a whole new cultivar. So, so the world is your oyster out there, and so hopefully this has inspired at least some people to go and take a closer look and maybe try their hand at making some crosses and doing some selections and having some fun with genetics.
When things clear up, we'll have to have you back either on here or for practicum where we can talk about maybe working with daylilies or irises or something like that. And
sure, absolutely.
I would talk about, because I know I have some friends that do work in that. My uncle used to cross irises and really enjoyed that. There's a lot of people get a lot of joy out of doing their own crosses and developing new plants. And like you said, some plants are much easier than others to do crosses and have new varieties develop. And I think people understand today the complexity of what goes into the experimentation crosses, that it takes many years of effort and multiple scientists working together to make these things come true, and field testing over years at the experiment station and then taking them out to individual farms to further test them. And we do that with the strawberries a lot. Mm-hmm. And it all comes together eventually, but it's a heck of a lot of work. So I give a lot of credit for, and also for the successes that you've had. And Pete, I don't know if you want to comment on anything. Pete Nietzsche's been with us answering questions, doing a great job. Peter, do you have any closing comments as well?
No, hopefully people learned a lot tonight, and hopefully you're trying some of our varieties and enjoying that. We released Scarlet Sunrise, and there was seeds available right around when the pandemic started. So we'll continue to make that available, and so hopefully more people will grow it next year. I think some of the growers put out transplants for sale, but I know gardeners have been snatching everything up this year to grow their stuff in their home gardens.
Yeah, gardening is very popular this year.
We've got so many inquiries from people that are getting out there for the first time. Even in my neighborhood, I see people gardening I've never seen garden before. Well, I want to thank Tom and Peter and everybody that helped out today, Rich Weidman, Dave Smella, and everybody that joined us today. We greatly appreciate you staying in tune for the whole hour and a half, and I hope to have you join us next week. And as I said, next week we're going to cover common problems that occur within your landscape and some strategic ways that are effective to control disease and insect problems and animals that may try to wander in and annihilate everything in your garden. So we'll give you some real practical solutions next week. Thank you, everybody. Good night.
Thank you. Good night.